If you are watching my videos in the order I made them in the playlist, you would probably already be realizing at this point, hey, drug molecules are so complicated because I could have an agonist, I could have an antagonist. Now, if I have an antagonist, is it pharmacologic? If it's pharmacologic, is it direct or indirect? If it's direct, is it competitive or non-competitive? And we have all these sorts of crazy diagrams that we have discussed so far. If it's now an agonist, is it full or partial? So many questions needed answers, and we try to answer them as far as the drug is concerned. But we haven't really paid that much attention to the possibility that there's also equally that much variation with regards to our receptors. And so, I would just like to discuss in brief, okay, the different types of receptors based on how many references classify them. Now, this is not a comprehensive discussion because if you want a more complete discussion of the types of receptors, you would better be off watching something related to cell biology or molecular biology. But if you just want something simple enough for us to use in our drug related discussions, then this may be enough. So, in many texts, receptors could be classified into four. So, those would be type 1, type 2, type 3, and type 4. So type 1 is for the ionotropic receptors, type 2 metabotropic, 3 kinase-linked, and 4 nuclear receptors. Sometimes you will see them being called differently, but I believe these are the most commonly used terms. First, let's start with ionotropic receptors, which from the name itself means that there's a relationship to ions here. Now, what you should know is that most receptors are actually transcellular, meaning our receptors are embedded in the cell membrane. So if I could probably draw a cell right here, and then you assume, okay, so these are my drug molecules out here floating. Where will these drugs interact? Where's the receptor? The receptors are actually just here, somewhere in the cell membrane, waiting for the drug to bind there, and then boom, we have an effect sooner or later. So let's say that this right here is the cell membrane, and then I did mention that I have this receptor, okay, this one in yellow, embedded in it. Let's assume that this space at the top is the outside part, extracellular environment, and then at the bottom is the cytoplasm or intracellular environment. So of course, since our drug molecules should be floating outside, let's assume that I have this green circle as the drug, and then this drug, or this ligand as we call it sometimes, could go to this active site of our receptor. Now, once a drug or a ligand binds into the proper active site of an ionotropic receptor, it will actually cause this receptor to open. So actually, most of our ionotropic receptors are channels or pumps. Though in this video, I don't intend to differentiate these two words. But channels or pumps are generally terms for proteins that, once they are activated, okay, or they are triggered by some ligand, in this case the drug, they will open. And this will cause this cavity to allow either the influx of some ions from outside to inside, or efflux of some ions from inside to outside. Of course, to know which is which, you have to study those receptors separately because we have different channels for different things. Sodium channels, calcium channels, potassium channels. If you're even hearing about those things before you watch this, that's that means you're gonna understand them better later on. But remember, why do we even need to know if ions are going in and out? Because remember, the ion movement to the inside or to the outside will cause some kind of effect. Just just to give you an idea, remember in physiology, we discuss action potentials, and we know that, for example, the influx of sodium from outside to inside will cause the depolarization and then will give us some kind of response. So basically, ionotropic receptors harness the capability of channels to allow changes in ion concentrations so that we will get some kind of effect we're looking for. And to know those specific effects, we have to deal with the individual ion channels one by one in the future. Next, We have type 2 receptors, which are called metabotropic receptors, and I think that is because we have to deal with different metabolites, and this means it's a little more complicated than type 1. In type 1, all we have to know is, oh, ions go in, oh, ions go out, and then we're done. We just need to assume what that means later on. But in type 2, it's kind of a story because there's a lot going on. And most of the time, metabotropic receptors are correlated almost every time to what we call as G proteins. Okay, G proteins because they have a certain affinity to GTP. Okay, but there's a lot more to that than how we define it. So just to give you a brief picture of that, let's again assume that this is a cell membrane right here. Okay, and then this is outside, and then bottom is the inside. Now, let's say that this is a receptor, a type 2 receptor, or a metabotropic receptor. Almost every time we discuss this in pharmacology, we assume that this is automatically linked to a G protein. It's not always the case, but it's safe to assume for basic discussion that it is. And the actual G protein, my friends, is not this. Many people are confused. This is not the G protein. The G protein is something here below or inside the cell that is only connected to this receptor. So this three colors right here with the alpha, beta, and gamma subunits comprise the trimeric, the three of them, the trimeric G protein. But the receptor here is only coupled to the actual G protein. That's why the proper term for this should be the GPCR, G protein coupled receptor, because that's what it is. It is only the receptor that serves as a messenger to the actual G protein that's waiting inside. Okay, so again, this is G protein couple receptor. This is the actual G protein. Now, by default, in the inactive state, a G protein is bound to a diphosphate, GDP. Okay, but let's assume that I have a drug molecule from the extracellular environment binding to the proper active site within this GPCR. Of course, that will cause changes in the structure of this receptor, which will trigger our G protein below to have GTP displace or get rid of GDP. So there's some kind of substitution reaction going on. The GTP inside the cell, okay, will displace this GTP. So GTP, goodbye. So we get rid of that. And then the GDP here is replaced by GTP. That is what happens once our drug binds to the receptor. So, like, the drug was just a messenger telling the G protein at the bottom to start working, you know. So, in that case, sometimes I could probably call the drug as the initial or the primary messenger because it's the one that keeps that makes everything start going. Okay, and then once I have GTP with my G protein instead of GDP, this will eventually cause the subunits to separate. Specifically, the alpha will be separated from the beta and gamma. So we will have alpha alone with the GTP, and then the beta and the gamma will stay as a pair. And then they will have their own separate ways. Like, hey, I'm alpha, I'm going to go somewhere. Hey, we're the beta gamma duo, we're going somewhere. But eventually, wherever this somewhere is, or whatever these question marks are, they will lead to the generation of what we call as secondary messengers. Secondary because remember, our primary messenger was the original drug molecule here. And it is actually the secondary messengers that will give us the effect we are looking for. So as you see, it's quite a long story. So just to reiterate the flow, again, we have a GPCR at the cell membrane. We have a primary messenger, which is just our drug molecule, binding to this receptor, triggering our G protein to become active, right? Have GTP, dissociate. And then, usually, once these subunits go to these question marks are usually enzymes. And it's the enzymes which actually give us our secondary messengers. And as I have mentioned, it's the secondary messengers which will give us the actual effect downstream or down the line. Now, for most cases, this is already enough. But I believe that if you want to really associate these G proteins to something understandable, especially when we get to autonomic drugs, I think it's necessary for you to know that the three most important G protein subtypes are GS, GI, and GQ. And I will be discussing them also briefly. First, The GS and GI are quite similar because this just stands for stimulatory and this one stands for inhibitory. And let's just assume that GS and GI are like contrary to each other or maybe complementary because they regulate each other, right? Because in both cases, whether it's GS or GI, their enzyme of focus, there's always an enzyme, right, for GPCRs or for G proteins. The enzyme of concern for GS and GI is both adenylate cyclase. And what adenylate cyclase does is it makes ATP cyclic so that ATP becomes what we call cyclic AMP. Makes sense, right? With the name. Or what probably many would be hearing as CAMP, CAMP. It's just that GS will promote that to happen, but GI will prevent that from happening, thus giving us the meaning of S and I. And camp is so important, and you will only really realize that if you go forward in my discussions, because camp gives us so much effects like bronchodilation. It will also have something to do with cardiac function and many more. Now, another G protein subtype is the GQ, wherein GQ is not linked to AC or adenylate cyclase, but instead linked to something else called phospholipase C. Now, if GQ stimulates the activity of phospholipase C, what phospholipase C will do is to act on phosphatidylinositol molecules. Usually, the phosphatidylinositol molecules are embedded in the cell membrane. And then, from the name lipase, it will break that down, right? Going back to your biochem enzyme nomenclature. So when you break down this phosphatidylinositol, you will encounter two important products. One of them is inositol phosphates. The most common is the triphosphate, so we abbreviate that as IP3. And then the other one, of course, this IP3 takes care of this part of the name. And then the remaining phosphatidyl will actually refer to diacyl glyceride, or sorry, it should be diacylglycerol or diglyceride. Though most of the time it's abbreviated as DAG or DAG. So we could say that GQ stimulates phospholipase C to produce the secondary messengers IP3 and DAG, which will give us the effects that we will be studying in the future. Okay, so for GS and GI, our secondary messenger is CAMP. For GQ, our secondary messengers are IP3 and DAG. And at the moment, it may be boring if you feel like, oh, do I really need to memorize this? You will appreciate this better moving forward. Next, type 3 receptors are kinase related. And remember, in biochemistry, when you say kinase, these are enzymes that allow the transfer of phosphate. Okay, so usually kinase-linked receptors deal with receptors in pairs. Because let's say I have two of these kinase-linked receptors, and then let's say one drug goes to one of the receptors and then another goes to the other receptor. Once these two receptors have been occupied, this actually serves as a trigger for these two separate. Remember, these are separate, right? This is separate from this one initially. But once the drugs have occupied both of these receptors, there will actually be dimerization, meaning these two will come together. And then after that, notice there are blue boxes at the bottom. These are actually kinases. Once the two receptors dimerize, the kinases start making their moves. So these kinases may phosphorylate each other, or they may actually transfer phosphate to other molecules or proteins going to them. And you may be like, oh, isn't that vague? And the answer to that is, if you actually go further in studying this in cell biology, there's just so much going on with these kinases at the bottom. But all you have to know is that the reason why we call them kinase linked is because in the first place, these receptors are really coupled to kinases at the bottom, very much similar to how in Metabotropic our GPCRs are coupled to the G proteins. Okay, and I think like if you feel like metabotropic receptors are already complicated with that flow, if I discuss the kinases here further, it might even equal or even exceed the level of complexity in this, and I don't think this is appropriate to discuss at this stage. Yeah, but with that said, let me skip this already and go to the last common receptor subtype, which is the nuclear receptor. Now, this is very special. Why? Because all you have seen here is that for type 1, type 2, and type 3, all of them, remember I'm saying this, all of them are transcellular, right? Because the receptors in type 1, in type 2, and type 3, as you can see, are embedded in our cell membranes. But in nuclear receptors, they are actually intracellular. Meaning, if I have my drugs here on the outside, and they need to bind to nuclear receptors, the nuclear receptor is actually here inside, and the drugs really need to go inside the cell to do their job. So, as you can see, for example, this is the cell membrane. Normally, for type 1, type 2, or type 3, the receptors will be found here. But for type 4, it's not, because the receptors are floating in the cytoplasm inside, and the drug has to do the job of penetrating or getting absorbed through the cell membrane so that the drug will encounter the receptor inside the cell. That's why intracellular is the term we use. Now, once the drug and the receptor interact inside, this complex between the two will allow them to go to the nucleus. That's why we call them nuclear receptors. And then change the structure of nucleic acids. Of course, usually it's DNA. And then, as you know, in the central dogma, DNA would control pretty much the encoding of everything. So many proteins could be changed by different genes, and so therefore the effect will only come out once we have controlled, modified, okay, the activity of the genes relying or lying inside the nucleus. And now, this is very important because if you see, evidently, the action of nuclear receptors is not as simple as the other ones. For type 1, type 2, and type 3, you just need to bind to the cell membrane, you're done. But since in type 4, you really have this waiting time wherein the receptor is here patiently waiting for the drug to go inside, there's this kind of lag time, like, oh, why am I not getting any effect? That's because for nuclear receptors, you have to wait for the drug to first go inside the cell membrane, and then another waiting period for the drug to go to the nucleus, and then another waiting period to wait for the DNA to be modified, the way it is expressed. Will it be more expressed? Will it be less expressed? There's a lot of waiting going on here. That's why it's not as immediate as the effects of type 1, 2, and 3 receptors. That's why for hormones, a lot of hormone receptors, especially steroids or nuclear, you don't expect their effects to come out immediately because of that said lag time. And I think I will discuss more about these when we go to steroid hormone pharmacology, but that would be a long road ahead. With that said, these are the four main types of receptors we will encounter.